very good evening to everyone. Am I audible? Are you able to hear me? Please confirm in the chat so that I can listen. I perfectly audible to all of you. Am I audible? Please confirm if I'm audible.
Am I audible? Am I audible now? Okay, please confirm, students, if I'm if it's perfectly audible. Apologies for the inconvenience. We'll start right away now. Today is going to be a very important topic from exam point of view also, and also from your learning point of view also. Okay, this is a continuation of biochemistry, structure and functions of protein and DNA, and along with that we will also study membrane structure and function. Okay, a very good evening to everyone. Yes, so let's start learning. So first we will discuss about proteins. Okay, now proteins, as we know, they are required in body's every functioning. They are most structurally and functionally diverse. You will get many type of proteins. What type of molecules are they? Biomolecules, means they are organic molecules only made up of carbon, hydrogen, oxygen. They have varied function, involved in almost everything that your day-to-day activities involve: metabolism, support, transport, regulation, motion. I'll show you some of the functions around each of these. So let's start with metabolism. Now, metabolism, one of the important aspect is enzymes, which will catalyze different reactions. They act as biological catalysts, and their function is to speed up the biological reaction. If you look at the enzyme, what exactly it will have an active site where the substrates are going to come and bind, and they are together going to form an enzyme substrate complex. Once the enzyme substrate complex forms, it is going to facilitate the formation of product, after which the product leaves. Like this, the enzyme doesn't undergo any change, but it will catalyze many reactions one after the other. These molecules are nothing but these are enzymes only. Okay, you have many enzymes you might have heard: lipase, amylase, lactase, protease, lot of enzymes which are used. Even in molecular biology, also, the ones you study: polymerase enzyme, DNA polymerase, ligase, which will join the phosphodiester bond between two broken ends of a DNA molecule. Even in industry, many enzymes are useful. So, they are nothing but protein molecules only. If you look at the protein molecule, you are seeing that there is a thread like structure. Right? So if you see, this is the thread-like structure, and thread-like structure, it is having different coils and different loops and everything, and that's how they form some specific structures. So you can see some sheet-like structure is there, some helix-like structure is there, some random loops, coils. This is what is called as a 3D conformation of the protein. Okay, this is called as 3D conformation of the protein. Now, support wise, what is the function? Yeah, they are structural proteins. Now, mainly these enzymes, they are serving as functional proteins. Their function, they will serve some function. Something will come and bind to them. Now, even if they are not enzymes, some receptor proteins, so they also are functional proteins where some ligand or something will come and bind with them. Okay, but these are structural proteins. That means they are present in some making of some structures. For example, in our carrot, in our hair, nails, then in animals, horns, hoofs, what is present is keratin protein. Okay, in the cells, inside the cell, we have cytoskeletal element. So the component is keratin. Collagen is there. It supports ligaments, tendons, skin, all that. Even the silk, which the spider cocoons will be doing. So these all are structural protein. Silk is made up of silk fibroin. Okay, then you have elastin also, which is present in the cell. It will be giving elasticity. It is present in abdominal walls, then lung walls, where stretchability is required, right? Something like that. Transport proteins, very important. You have channel carrier proteins. They are going to have function in allowing substances to enter and exit the cell. They are present in the plasma membrane. We'll talk about different types of channel proteins. And then another one is transport. So what they will transport, they will actually bind with some molecule, and they will allow to take it from one place to the other. For example, hemoglobin. In RBC, this is a hemoglobin protein. Okay, so its function is to bind oxygen. It will bind the oxygen and it will deliver it to from one tissue to another tissue. Okay, then you have defense like antibodies. We know immunology a little bit. That is, they will combat bacteria and viruses. Right? We have that immunoglobulin, which is nothing but the antibody. They have heavy chain, light chain. What are these? These are nothing but protein molecules. Okay, they are made up of beta sheets like this. They are made up of some domains. So this is one polypeptide chain. This is one polypeptide chain. Okay, different immunological functions these antibodies have. Fine. Next is regulation. So regulation works in hormones and in receptor protein. Hormones, what are they? We have hormones throughout the body. They act as messengers, they act as signals, okay, that affect metabolism in different function. For example, the signal from a neuron to a muscle, right? And also insulin, the regulation of amount of glucose in the blood and in other cells also. Human growth hormone, its presence determines the height of an individual. So all these signalings, they will be coming, and on the surface there will be something called as a receptor protein. So receptor protein built into the membranes of nerve cells. They will detect chemical signals. Chemical signals, they will act as neurotransmitters released by other nerve cells, right? So see, this is what is called as a ligand. Ligand is what is a primary messenger. Primary messenger binds with the receptor. Receptor receives it. Generally, receptor is present in this plasma membrane, and the receptor will in turn give the signals to secondary messengers. Okay, then they will have the effect on the nucleic acid. That is, the change in the gene expression will be observed. Fine. Next is motion. So if you know about muscle contraction, then we have the um, we have the actin and myosin which slide along each other. So they make up the muscle fibers, and those contraction only leads to the motion of the muscle, right? Even the motor proteins, they will have some heavy chain and some light chain. So you can see that how motor proteins are carrying these transport vesicles. Okay, so they can move along the microtubules. They are carrying these transport vesicles, and that way they are able to move through it. Fine. Apart from that, you have flagella, which moves through the cell. and means which will help in the movement through the cell. And cilia, they will move the contents around the cell, right? So if any particular, let's say, sperm cell movement, then they will have this flagella. With that flagella, they will be able to swim forward towards the egg. Or else the cilia which are present in the lung epithelium, cell, air pathway epithelium. So there they will have some power stroke, recovery stroke. So that way they will beat off the bacteria and keep it at bay. So what are those? Those are nothing but proteins only. Okay, they will be consisting of some protein molecules, like microtubules are made up of protofilaments, right? And these are made up of actin, myosin. What are these? These are nothing but proteins only. Now let's move to the structure. That was something about the function. Protein structure, the monomers are called as amino acids. Okay, here are some examples of proteins: growth hormones, hemoglobin, rubisco, just to show you how different protein structures are. So the structure is actually made up of proteins are made up of individual unit building blocks, which are amino acids. So whatever proteins we have in our body, they are mostly made up of 20 standard amino acids. Okay, they will be standard amino acids because most of the proteins will have these 20 amino acids only. So if you remember in the first slide, I told you that proteins are diverse. Now why are they diverse? Because there will be so many possibilities with these 20 amino acids. Just to take example, you take the alphabet set. You have 26 letters. But with 26 letters, you can make so many words. There is no end to it. And depending on the sequence of the letters, the pronunciation will be different, the word meaning is different, how you use it in different sentences will be different, the way it appears is different. Same goes with proteins also. That's why they are diverse, okay? Different. Out of the 20, you should remember 12 are made by the body, and the rest 8 you have to supplement. That is from the food. They cannot be produced in the body. That's why they are called as essential amino acids. Okay, so you must know the list of essential amino acids also. You must learn from the books. I'm not discussing here because of time constraints. I have to cover many topics today. So, polymer, what is polymer? Protein is actually a polymer. Poly means many, mer means molecule. So, it's consisting of many molecules. And it's also called as a polypeptide. Peptide, why? Because the amino acids are joined by peptide bonds. Okay, many bonds. Protein can be one or more polypeptide chains folded and bonded together. So as you can see here, hemoglobin actually has 4 chains, 4 polypeptide chains given in different colors, and each one of them has folded in different way. Okay, so in this protein they have many structures. They are not visible properly. They have just given an overall structure, but there are many detailed foldings. So each of these folded proteins they have assembled together, and that's how they have got a complex shape. Okay, so some proteins will have quite large and complex molecules, and overall they will have a 3D shape, three dimensional tertiary structure they will have. We'll discuss about the conformations. So talking about amino acids, what is the structure of amino acid? There will be a central carbon alpha. So in the center, this molecule amino acids with which proteins are made up of. So they have this central carbon alpha. Okay, carbon has 4 bonds, 4 valency. So with one, there will be an amino group. So as you can see, this is the amino group. That's why they are amino. And another one will be acidic group. That's why they are called as amino acid. Okay. Another group will be R group. So anyways, one of that is hydrogen. Okay. One will be hydrogen. And another one will be R group. This R group is called as a side chain. So this is also called as a variable group because in different amino acids, these groups will be different. And this is what confers unique chemical properties of the amino acid. Okay, so depending on what type of amino acid is there in the protein, the behavior of the protein, the function of the protein, the structure of the protein, all will be affected. Some R groups are very big, some R groups are very small. So that's why we will take an average molecular weight for this overall amino acid. Okay, because rest are same. It's just that the R group molecular weight will be different. So just remember, average molecular weight of an amino acid is somewhere around 120 Dalton, 128 Dalton. Okay, but this is individual amino acid. When we take in a protein, then it is different. Okay. Okay, so Jaya Sai Ramya, selenocysteine and pyrrolysine are not standard amino acids. Okay, they are 21st and 22nd, but officially they are not yet in the standard list. Okay, because you will not find them in all proteins. If you see the rest 20, you will find them in all proteins. But selenocysteine and pyrrolysine, they are special amino acids. That too, they are incorporated through a special mechanism. Okay, we know that amino acids get incorporated into protein through the process of translation, right? Where there will be specific tRNAs for specific amino acids. But these will not have their own tRNA coding ones, but they will be specially incorporated ones in the protein. Clear? So we will have to know about the amino acids. What type of amino acids are there? Classification. So there are many classifications. One, as I told you, essential, non-essential, that you can just study from your books, BSc books, right? And another one is glucogenic, ketogenic. That is not on your level. It will be when you are going to MSc level. But the most important one, which you need to know, is non-polar and polar. This is the main category. Okay. One is non-polar amino acid, another one is polar amino acid. Now, on what basis we are calling something as non-polar and something as polar? So, when we have bonds such as OH or NH, right? So, if you see the reacting elements, oxygen is more electronegative, means capacity of attracting electron is more. Hydrogen is less electronegative. Nitrogen is high electronegative, hydrogen is comparatively less. So, whatever electrons they are sharing in between them, those are mostly attracted towards the oxygen and towards the nitrogen. Got it? So that's why their electron cloud is slightly distorted. Got it? The OH bonds, then the NH bonds. So as a result, what happens? The one towards where the electronegativity is higher, that side you will get a delta negative charge, a partial negative charge you will get. And the other side you will get a partial positive charge. Okay? Even here also, nitrogen will have partial negative and this will have partial positive. Got it? So if you take water molecule we know that it is consisting of oxygen joined with two hydrogen, right? So oxygen is more electronegative, so this side will be delta negative, this side will be delta positive, means this end of the molecule will be delta positive. Now, if you have any other molecule which is having also some delta positive and delta negative ends, let's say you have a NH group. So now what will happen? This delta positive end is going to attract the delta negative end. There will be some electrostatic attraction between these two. So they will interact with each other. Okay, they will interact with each other. So this is what is called as a polar molecule because they have polarity. You can see one side is negative, one side is positive. So similarly, this is also polar molecule. So if polar interact, they will be able to interact. There will be interaction between them. Okay. But what if we take non-polar? So what are the non-polar components? CH bond, CC bond. Okay, these are non-polar components. That means they don't have any polarity. They are, if you take a CH bond, the electronegativities are almost same. So both pull the electrons with almost equal affinity. So there is no polarity. Electrons are equally distributed over the molecule. So delta 0, delta 0. Now you imagine a non-polar molecule is trying to interact with a water molecule which is polar. So how can a non-polar molecule, in short you can say this is uncharged. This is not having any partial charges. How can an uncharged molecule interact with a charged molecule? Is that possible? Tell me. Can a non-polar molecule interact with a polar molecule? Polar molecule which is having partial charges. Can it interact with something which has no charges at all? So here they were interacting, the delta positive end was interacting with the delta negative ends. But here, can the delta positive end interact with delta zero end? Is that possible? Think about it, students. Because here there was electrostatic attraction. Positive and negative charges, opposite charges, they were getting attracted. But here there are no charges. Isn't it? Is it? Are you following, all of you? Are you following what I'm trying to say? Please confirm. So what I'm trying to say is like charges, like repels like. Okay? Like repels like and like attracts like. So that means what? The non-polar molecules, they will interact with non-polar molecule. Polar molecules, they will interact with polar molecules. Okay? Right? Now, what does hydro mean? Hydro means water. What does phobic means? Phobic means fear. So if I say hydrophobic, what does it mean? That these molecules cannot interact with water. So that makes sense because they are non-polar and water is polar. So polar and non-polar, they cannot interact. So that's why these all molecules, they are hydrophobic. They cannot interact with water. Okay. You have to memorize these: glycine, alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, and tryptophan only to some extent, just because it has this aromatic rings. Okay, tyrosine and tryptophan to some extent, so I'll put a star over here because it is slightly polar. Because of this NH group, no, it is slightly polar. But rest all side chains, if you observe, glycine is the smallest amino acid, so it has just a hydrogen in the side chain. Alanine is only having CH3 in the side chain. So if you see all the side chains of these non-polar amino acids, they are having CC and CH bonds. Either CC bonds or they are having CH bonds. Okay. Methionine also you can see CC-CH bond. CS bond is there, which is also a non-polar bond. Yeah, but if you have SH, that is a polar bond. Okay. But here you don't have SH, you have S. That's why this is non-polar. And anyways, aromatic ring, that will be considered as non-polar. Okay, hydrophobic. And proline has a very characteristic type of side chain. You will see that the side chain is consisting of 3 carbons, 1, 2, 3. But then the last carbon is joined with its own amino group. So remember, this special type of structure of proline is called as an imino group. Okay, it has a cyclic structure, not aromatic. I hope you know the difference. Aromatic will have the property of resonance. They will have some alternating pi bonds for resonating. But here you don't have any resonance. This is not aromatic. This is just a cyclic molecule. Got it? And glycine is the smallest one with only hydrogen. Sulfur containing amino acid is methionine. So these are actually important. Then coming to polar charged and hydrophilic. So some are charged, some are uncharged. For example, all these are uncharged, but then they are considered as polar. Okay. Tyrosine, again I will put a star over here because owing to its aromatic ring, it is slightly non-polar and slightly polar because of the OH group. Okay. Then you have serine, threonine, all these have OH groups. You can see they are giving polarity. Because of this OH groups only they are polar. Cysteine is polar, SH group. Amino group in asparagine. Not the one which is present as in the main chain. This is in the side chain. Even this one present in the side chain. Okay. And then there are some acidic amino acids which have an additional carboxyl group in the side chain. Then here also glutamic acid, that also has a side chain carboxyl. Basic amino acids, they have extra amino group in the side chain. So you can see extra amino groups in the side chain. That is lysine, arginine, histidine. Okay, so all these names and the category, most important is just the category. You try to remember because they will affect the protein structure, how the protein is going to behave. Okay. Now, how do you build proteins? We build proteins by peptide bonds. So you have this amino acid, one amino acid. You have another amino acid. Now the OH of one carboxylic group joins with the AH of another amino group. So one OH and H comes out as water molecule. And then what will join together? CO and NH will join together. So this is what leads to the formation of peptide bond. Since water comes out, what is this called as? This is called as dehydration synthesis, which means that D means removal of hydration, removal of water. Okay. linking amino group of one amino acid to carboxyl group of another amino acid. And this is what results in a CN bond. This type of bond is called as an amide bond. Okay, this is called as an amide linkage. Chemical functional group name is amide. Just because it's about protein, then this is a special name given called as peptide bond. Another important point about protein is that we always write the protein from N to C. Okay, like how DNA RNA we write from 5 dash to 3 dash always. So like that here we will write always N to C, C terminus. And if you remember, I told you individual amino acids they have 128 Dalton. But then when they are involved in the protein, one water molecule comes out of them. Water molecule molecular weight is 18. So 128 minus 18, that means in the polypeptide chain, the amino acid molecular weight is only 110 Dalton because one water molecule will be lost from each amino acid except the terminal ones, because from terminal ones only one end will be involved, the other end will be free. So anyways you will have a free amino group and a free carboxyl group at the end. Okay. Now coming to the protein structure and function. The function depends on structure. Whatever the protein function will be there, it depends on the structure of the protein, especially how it is appearing in 3D, tertiary structure. Whether it is twisted or whether it is coiled into unique shape. So this will be depending on such things. Okay. Talking about the primary structure, there is a conformation of the protein, how the proteins will be folded step by step. So primary structure, it is the order of amino acids in chain. So something like this. You can see this is a polypeptide chain. We start with the amino group and then you can see it's the first amino acid, second, third, fourth, fifth, sixth, like that it is continuing. So this entire polypeptide chain ending in carboxyl group. So linking each amino acid there will be peptide bonds. Linking each amino acids there will be peptide bond. So this is what is the primary structure. Okay, the linear structure. So there are some keywords to learn over here. It is the linear structure. It is just a straight one. Amino acid sequence, and this is nothing but what is coming from the gene, from the DNA. DNA is made into mRNA, and after splicing and all, whatever the genes will be present, whatever codon will be there, based on that the protein will be made. Okay. Slight change in amino acid sequence can affect protein structure and its function. Even just one amino acid change can make all the difference, right? So we know how the gene mutation can have an effect. So a slight small mutation can also have a big effect on the function of the protein. Why? Because the change in the gene will have a direct impact on what amino acids are getting encoded. And if amino acids are different, the interaction between the amino acids will be different, the conformation and structure of the protein will be different, and obviously the function also will be different. Okay. Next, coming to the secondary structure. So this is more of a local folding along short sections of polypeptide chain. Now, very important point to remember, this is where the interaction between amino acid means the peptide bonds will be taking place. Okay, peptide bonds, right? So this is a connection, hydrogen bonds between backbones. Okay, only the peptide bonds will interact. For example, you can see this is a linear polypeptide chain. You are able to see linear polypeptide chain. Here also linear polypeptide chain. Then this is only folded back, another linear polypeptide. You can also see the peptide bonds. I hope you understand CONH. I just showed you. So you can see here CONH. So and you can see there is a bond between them. Between the two peptide bonds, there is something called as hydrogen bond. Okay. So secondary structure is actually stabilized by these hydrogen bonds. Right? Different type of secondary structures are there. That is alpha helix, beta pleated sheet, right? And sometimes, yeah, if you have the functional proteins which I mentioned, enzymes and proper proteins, those are called as globular proteins. Okay, because they end up in some globule like structure which I showed you before, something like this, right? These are globular proteins. This is an enzyme showing the active site. This is also globular protein. They are globule, like spherical shaped in structure. But comparatively, you can see collagen, they are straight, linear structure, longitudinal structures. Okay. So this is what you have, the collagen, fibrous protein, which will have only secondary structure. They don't have primary, sorry, they don't have tertiary, quaternary. They have primary, and from primary they have secondary. Okay. So this is what you see in proteins, proper conformation, ribbon structures. These are alpha helices components. These are sheet components. Sheet means they are arranged linearly like this. Okay, like this they are arranged linearly. Helix means they are having some helical appearance like this. But what is main important thing to know over here, that they are stabilized by hydrogen bonds. Okay, secondary structures are stabilized by hydrogen bonds between what? Between peptide molecules. Okay, between peptide molecules. Peptide molecules. Peptide bonds rather. So more precisely, I will write over here peptide bonds. Remember that. Okay. Very important point, that in secondary structure it has got nothing to do with R group, because these carbons will have some R groups also. So these R groups, they actually point above and below. Okay, they come above and below the plane. This is like a sheet. So you will have a upper sheet, this you have a lower sheet. So they are like up and below. Yeah, what is that? 1 to 13. What is that, Manik? I didn't get that what you are saying. So even here also you will see one peptide bond. So here also there is the peptide bond. So between two peptide bond there is hydrogen bond. No, no, no. There is nothing called beta helix, Manik. You have either beta sheet, beta pleated sheet. There's a pleated appearance, that's why, or you will have alpha helix. Okay. Yeah, helices are of different types, and beta name is also associated with different other structure. For example, there is something called as a beta turn, but that is not a helix. Okay, Manik. Beta turn is consisting of 4 amino acids. It's an existing chain, means continuation. So that chain, 4 amino acids, there will be bonding between them, and this will exist as a turn. So that is called as a beta turn, right? Beta turn or beta loop, like these names can be there. And helices also, there are different types of helices. I will not go into details of all that today, right? And as I mentioned, there will be hydrogen bond. So you can see between beta sheets you have this hydrogen bonds, right? In alpha helix you have this hydrogen bonds. Now between different parts of the protein in tertiary structure, you will notice that there is one amino acid, another amino acid, both of them are non-polar. If you remember from the classification, isoleucine, valine, both are non-polar. So two non-polar groups, they will stick together through hydrophobic interaction. Now hydrophobic means what? Fear of water. As I told you, they will be not able to interact with water. So when a protein folds in aqueous environment, aqueous means in water environment. So then they will try to escape from the water and they will accumulate together. If you have a positive charge groups, some basic amino acid, if you have some negative charge groups, some acidic amino acid, they will stay together. So they will stay together with some electrostatic attraction. So you can see some electrostatic attraction is there. Sometimes some metal ions will join some parts of the protein together. So all this lead to a complex conformation of the protein. Something like this. Now further, if there is another folding level, that will be tertiary, third level structure. Okay, so it's not necessary that they form in the exact order, that is first primary, then secondary, then tertiary. But yeah, obviously first the polypeptide chain will be made. Then the local structures will be aligned, that is secondary structures. Then the tertiary structures will be made. So tertiary structures are made between different parts of the protein. For example, you have let's say alpha helix, then let's say some turns are there, some loops are there. Okay, so from there the R groups are coming out. Here is one R group, variable group, which is consisting of OH group. Then from here also one more R group is coming out. Because we know that in secondary structure there is no interaction with R groups. Okay. So whole molecule folding is what is tertiary structure. Folding of the entire polypeptide chain. Here different types of bonds can be present. You can have hydrogen bond between OH group and oxygen of another one. You can have hydrophobic interaction where hydrophobic groups they will cluster together to stay away from water. Van der Waals interactions can also be there. Disulfide linkages are there between two cysteine residues. Okay, cysteine, as I showed you, it is having SH group. SH is sulfhydryl group. So two sulfhydryl groups, hydrogen will come out from them. That is, oxidation will happen, and these two will link together, forming a disulfide bridge. So this can bring two different parts of the protein, okay, or it can bring two different proteins also. If you draw immunoglobulins, if you remember, they are joined by SH linkage, right? Even the light chains they are joined by SS linkage. So these are proteins only which are joined by this such kind of linkages. Plus, you can have ionic bond electrostatic attraction. Positive, negative interactions can be there, right? So these are determined by interactions between the R groups. The hydrophobic interactions, which are anchored by disulfide bridges, ionic bonds are there, hydrogen bonds are there, and van der Waals forces. Okay, so these are globular spherical proteins which have tertiary structure. As I mentioned, secondary structure, fibrous proteins, they can have only secondary structure, but globular proteins definitely they need to have some 3D structure for their proper function. Okay. Last is quaternary structure. So two or more tertiary folded peptide subunit, they will be bonded together to make a functional protein. So this is where tertiary structures which are already made, they will come together and assemble in such kind of structures. Okay. Like you have hemoglobin, collagen. In hemoglobin, 4 polypeptides join. In collagen, 3 polypeptides join. But these are already in the folded form. It's not that these different polypeptides fold with each other. No, they are already in folded form. They just assemble together. So quaternary level of folding is only assembly between the subunits. So this will be called as these individual units will be called subunits. Overall, the protein will be an entire unit. Okay. So this is the overview protein structure. We start with a polypeptide chain. Then some parts of the polypeptide chain locally they will arrange into some folds, repetitive folds like alpha helix or some beta sheet. Such kind of secondary structures, the R groups will come out of them without interacting, but in tertiary structure, those R groups will interact between different secondary structures. So there will be a complex interaction between different parts of the protein that will give a overall 3D structure. And if several such tertiary proteins come together and assemble to form a more complex structure, that will be quaternary. Not all proteins will be quaternary. Like hemoglobin, it has 4 subunits. DNA polymer, RNA polymerase has 5 subunits, right? But there are proteins which are only having tertiary structure. They don't have any other subunit, like cytochrome, lysozyme, right? Ribonuclease. These are individual monomeric proteins. So they don't have any other subunits. But RNA polymerase, as we know, we need different subunits for different functioning. So that's why we need different subunits. Okay. So one very important part to fold the protein, we are folding in aqueous environment. As I mentioned to you that we fold in aqueous environment. Okay. Presence of water. Now here the polar and non-polar thing comes in picture. So polar groups are present on the surface. Polar groups will be more towards the surface while folding because they can interact with the nearby water. But the non-polar groups they will be mostly buried in the core. Okay, non-polar groups will be mostly buried inside the core of the protein. But the polar molecules they will be mostly present towards the surface of the protein. Clear? So this is what affects the protein folding. The most dominant type of folding in tertiary structure is hydrophobic interaction, by which the nonpolar molecules will assemble together. They will come together. Okay. So as I told you, the secondary structure is determined by hydrogen bond. Primary sequence is determined by the DNA genetic code. Tertiary structure is stabilized by hydrophobic interactions mainly, apart from the disulfide bridges, van der Waals forces, ionic bonds. And these multiple polypeptides also same, almost same. Okay, same type of bonds. There is something which is what is called denaturing a protein, in which the protein is properly folded and then it will have the proper function. Such a protein which will give the function it's called as a native protein. Okay, the one in which it will be having the function and in which it will be properly oriented, that will be native structure. But in case there is some denature and some adverse conditions, pH, temperature, or any chemicals, okay, then the 3D structure can be broken. Because if you increase the temperature, for example, temperature contains heat energy. With heat energy, there are some weak bonds which are stabilizing the protein structure. Hydrogen bonds, then van der Waals interaction, electrostatic, all these are weak bonds. Only the disulfide linkage is covalent bond and peptide bond is covalent bond. Rest all are non-covalent interaction. So that's why those will be broken. Okay. So if they are broken, then the entire structure is denatured. That means it is completely opened up. Okay, it unravels or denatures the protein. It will disrupt the different bonds and obviously it will destroy the functionality also. The function of the protein will no longer be there. So that was about proteins. Let's go to nucleic acids. Now, what is the function of nucleic acid? Yes, it serves as genetic material, stores different information. So in the cell inside the nucleus, even your this mitochondria, chloroplast also have their own genome. Okay, so they will have this chromosomes. Nucleus will have chromosomes. In that, genes are there. In the genes, the information is stored. What type of proteins it will make. So they are blueprint for building protein. Based on that information, only proteins will be made up. Okay. So what is the sequence? From the DNA you get RNA to proteins. This is central dogma of molecular biology. DNA to RNA is replication, DNA to RNA is transcription, RNA to protein is translation. Okay. So whatever DNA we have, the different bases that we have, so that is only incorporated into protein. Depending on whatever basis, so we have codon, triplet codon. Three bases will be coding for one codon. Okay, and three codons, three means three bases like that. One codon will be coding for one amino acid. So triplet codon, that's what we called. Okay, so depending on whatever codon is there, accordingly the amino acids will be incorporated. So it directly affects the structure and function of the protein. Got it? It transfers information which acts as a blueprint for the newer cells. Fine. So with that only, this information is getting passed on to the next generations also. So even it is for the blueprint for the next generation. Right? Now coming to the building block of nucleic acids. As I told you, amino acids are building block for proteins. So for DNA, RNA, these are what are nucleic acids. DNA, RNA, these are nucleic acids. So this is what is for nucleotides. Okay, these are long thread-like polymers. So yeah, they are linear structures, double helical. RNA is mostly single stranded, double stranded is also there, and they are made up of a linear array sequence of monomers called as nucleotides. So what does a nucleotide look like? It has actually three components. One is a nitrogen heterocyclic base. That's called as nitrogenous base. So this is what is nitrogenous base. It is two types. One is purine, one is pyrimidine. The reason these are bases because they have a huge content of amino groups, okay, NH3 groups, and they are rich in amino groups. And nitrogen because yeah, they are containing nitrogen, rich content of nitrogen. Apart from that, you have a pentose sugar. So pentose sugar is also called as ribose sugar. Ribose sugar means it's a 5 carbon sugar. So you can see here 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. Okay. In the nucleotide, this sugar carbons they are labeled as 1 dash, 2 dash, 3 dash, 4 dash, 5 dash. Dashed are given. Okay. Normal sugars it's not, but in ribose, in nucleotide, it is given dash. Why? Because purines and pyrimidines will also have carbon, hydrogen, and all that. So there you will have 1, 2, 3, 4 numbering. So here you have in the same molecule, if you have the same numbers, it will be confusing. So for purine pyrimidines, you have 1, 2, 3, 4 numbering. For ribose sugar, you have 1 dash, 2 dash like that. If you will notice that the purine and the pyrimidine base is connected with which carbon over here? The first carbon. Okay, that is one dashed carbon. The nitrogenous base is connected with that. This bond which is connecting both of them, it is called as glycosidic linkage. Just because a sugar bond is there, no, means a sugar is involved in the bond. So that type of bond is called as glycosidic linkage. Remember that. Okay, and the bond between, and yeah, there is another component which is the phosphate. So phosphate is joined with the sugar, that is at the fifth position carbon, it is joined with the phosphate group. This bond is what is called as phosphomonoester. Okay, this is monoester because this is what is a nucleotide monophosphate. This is nucleotide monophosphate. Mono means single phosphate. Okay, yeah, if you have triphosphate, two more phosphates if it is attached, then we will call it nucleotide triphosphate. Tri means three. So that time these bonds between the two phosphates, they will be known as phosphoanhydride bonds. You must know these bonds, okay? Sometimes they ask questions. So phosphoanhydride bonds, these are. But this one is between the sugar and the phosphate, it is phosphomonoester. But when the replication happens, this is cut off. The two phosphates come out, and the last phosphate is joined with another sugar from another nucleotide. Okay. Now this phosphate is joined with two sugars, one at this end, one at this end. So this bond is ester, the one with which it joins sugar. I told you it is monoester. But since this phosphate is now connected to two sugar, now this will be known as phosphodiester bond. Now it will be called as phosphodiester bond. Di means two. Okay. So only when the phosphate connects two nucleotides, that is in the DNA RNA, then only this bond is phosphodiester, which is present in the backbone of the DNA. Else, if you talk in a single nucleotide, it will be called as monoester. Fine. Now coming to pentose sugars, that is the ribose sugar, as I mentioned. So there are two related pentose sugar, one which contains the ribose, another one which contains deoxyribose. Now deoxy, from where the oxygen is missing, this is a regular ribose. At the second position, if you notice that there is a oxygen, okay, OH group is present. But here at the second position, you will notice that oxygen is missing. So this is what is called as a deoxyribose. DNA contains deoxyribose. The sugars have their carbon atoms numbered with primes, as I told you, to distinguish from the nitrogen bases. And ribonucleotides have a 2 dashed OH, whereas deoxyribonucleotides, they have a 2 dashed H only. And because here also you can get a phosphate, that's why we write 5 dashed phosphate end. And this side we have a 3 dashed OH, so that's why we write it 3 dash OH end. Remember, to attach one nucleotide, your 3 dashed OH end should be free. Okay, the 3 dashed OH end must be free. So what are the type of nucleotides that we have? We have two types, different nitrogenous bases will be there. First type is purines. Remember, purines are double ring. You can see one ring over here, another ring over here, right? And then you have pyrimidines. So under purines, what you have? You have adenine and you have guanine. Both are purines. Next is pyrimidines. Pyrimidines have single base, single ring base, nitrogenous base. Okay, single one. So there are three types: cytosine, thymine, and uracil. Okay, uracil is present in RNA, and DNA is thymine is present in DNA. Okay, so you have to means ideally you should know the structures, but if you have any problem, you can just remember, like you can draw somewhere and you can post it. Like you can remember what are the special groups. Else the rest of the skeleton is same. So you can see here, these are the special groups. Rest of the skeleton will be same. Yeah. Yeah, you want to know the numbering, Ajit? Okay. So numbering of purine pyrimidines. If you have purines, so you have this purines. Let me draw a purine over here, right? I have less time actually. That's why I was not showing you that. But since you asked, so these are all carbons, right? So this is nitrogen, this is carbon. Now for purines, you start the numbering from the side nitrogen. Okay. from the side nitrogen. Then you come to the nearest nitrogen. So nearest nitrogen is which one? This one. So next one will be 2. So you come this side. Okay. Then 3. Then 4. Then 5. Then 6. Then you go to the next one. 7, 8, 9. And this 9 is connected with the sugar. That is the first carbon what I showed you. No? This is connected with the first carbon of the sugar. For purines, remember the connection is C1 dashed to N9. Okay. And for pyrimidines, what is the numbering? So pyrimidine numbering, it begins from the bottom nitrogen. See, skeleton is same. 2 nitrogen like this and rest is by carbon. Okay. So here we start the numbering from the bottom nitrogen. And then we go towards the nearest nitrogen, which is this. So according to this side, it will be 2, 3, 4, 5, 6. Clear? So here the only option to connect with sugar is the first one only. So here first one connects with first one. That is C1 dashed connects with N1. Okay. Here C1 dashed connects with N9. Clear now, Ajit? This is what is the numbering. Okay. Now nucleic polymer, the backbone sugar to phosphate group. This is the backbone. So when many nucleotides join together, you will see a phosphate group, a sugar, phosphate, sugar, phosphate, sugar. So that's why we call it sugar phosphate backbone. Okay. Bases you can see they are coming out of the backbone. Okay. So here also there will be another strand. This is a primary structure. If you want a secondary structure, you will have another strand. So from there also bases will come out. And then bases will have hydrogen bond between each other. So that's the main stabilizing force between DNA. The bases will have hydrogen bonding. Got it? So what is this connection? As I told you, between two nucleotides you will have phosphodiester. This is one nucleotide, this is another nucleotide. Between these two is the phosphodiester bond. Again phosphodiester, like that. Okay. So new base is added to the sugar of the previous base. To the sugar of the previous base, the new base is added. So always the attachment of new nucleotide, for that we need a 3 dashed free 3 dashed OH end. Without free 3 dashed OH end, the nucleophilic attack cannot take place and it cannot attach. Okay, and polymer always grows in one direction, that is attachment is always towards the 3 dashed end. So always it goes from 5 dash to 3 dash direction. Remember that. Okay, and nitrogenous bases hang off the sugar phosphate backbone. So it's like a ladder, right? This is uh, one stand of the ladder, another stand of the ladder comes like this, and then they have hydrogen bond and then they are twisted also. So that's where it gets a spiral structure. The DNA structure gets a spiral shape. Fine. So putting the DNA together, backbone together, you have to see the 3 dashed end what I mentioned. So you can see at the 3 dashed OH end, instead of that, the phosphate of the new nucleotide is attacking over there. Okay. So instead of this sugar OH group, now you have the phosphate group of the next one which is attached, right? So the last trailing carbon, it will have free 3 dashed OH end, and the first one it will have a 5 dashed phosphate end. So always the growing is from 5 dash to 3 dash. So remember these 2 points. Protein always we write from N to C, and in fact translation also happens from N to C direction, and DNA synthesis always happens from 5 dash to 3 dash direction. Okay. So now coming to bonding in DNA. Now bonding in DNA, we have thymine, which is pyrimidine, and we have adenine, which is purine. So always purine bonds with pyrimidine. Similarly here G, which is purine, C, which is pyrimidine, they will bind together. But the binding is little bit different, in the sense they are both hydrogen bonds, but A and T, they are joined by 2 hydrogen bonds. Whereas G and C, they are joined by 3 hydrogen bonds. So you have to remember this, okay? Now, what type of bonds are these hydrogen bonds? They are actually weak bonds, okay, because they are non covalent forces. There is a weak interaction between the molecules, okay, and the DNA composition can be different. You don't need to have equal amount of AT and equal amount of GC. So, how much ever AT are there in the DNA, Okay, it's not that equal amount of GC also has to be there. Not That's not necessary. You can have more of AT bonds in the DNA, or you can have more of GC bond in the DNA. So don't forget that these are stabilized by two hydrogen bond. That means these are weaker. These bonds are weaker. Okay, than the GC bonds, these bonds are weaker. These are more stronger bonds, GC bonds. Because to break them, you require more force. Three hydrogen bonds are required, no? So you require more force to break them. So that's why if any molecule GC content is more, if any molecule GC content is more, we will say that molecule is more stable and the melting temperature will be higher. For molecules, Tm will be higher, melting temperature, because more effort and energy is required to separate the two bonds, two strands. So this is one strand and this is another strand. Another point, in secondary structure of DNA, the strands go anti-parallel to each other. Okay, and DNA is negatively charged. So you can see that the phosphate group, even after bonding with two sugars, okay, even after bonding with two sugars, the phosphate still able to retain a negative charge. That's why phosphates are present with DNA. They actually stabilize DNA and they also protect the DNA, because this negative charge will prevent the DNA from getting hydrolyzed by water molecules. Okay, so this is what is covalent phosphodiester bond, and this is a secondary structure of DNA. So the shape is double helix. It's present in helical structure. You can see it's like ladder only, which is twisted. Now the Watson-Crick gave this bDNA model. There are many forms of DNA, ABZ DNA. Again, because of time constraint, I'm not going into detail. Now this is one turn of the DNA. Okay, one turn. So this is what is the pitch of the DNA. Pitch means the length of one turn, rise per turn. So how much is the pitch? 3.4 nanometer. You must remember this. And what is the rise per base pair? So here is one base pair, another base pair. What is the rise between them? What is the length? So in each of the turns, there are roughly 10 bases. Okay, each turn contains roughly 10 bases. So total turn length divided by 10, then each one will be 0.34 nanometer. You can also represent them as Armstrong. So it will become 3.4 Armstrong, and here it will become 34 Armstrong, because 1 nanometer is equal to 10 Armstrong. Okay, and the diameter of the DNA is 2 nanometer. So you can see one strand is 1 nanometer. So another one 2 nanometer. 2 nanometer is the diameter. Okay. Hydrogen bond between the bases will be present. So why are hydrogen bonds important? Because they are the reasons, and yes, weak bonds are helpful. Why? Because it's because of the weak bonds only that the DNA is able to unfold. So when replication and all happens, they will open up a replication fork, if you remember, right? So the replication fork, it is very easy to unzip the DNA. That's why hydrogen bonds are very important, because overall, when so many hydrogen bonds are formed, it provides a stabilizing force also. It's the combined effect of the weak bonds that overall gives the strong structure. And apart from that, even it is easy to break with little bit of effort. So any enzymatic treatment can even break them very easily. Fine. So three differences between DNA and RNA. DNA has deoxyribose as sugar, double strand, double helix, and it has thymine as base. Whereas RNA, it has ribose as sugar instead of deoxyribose, generally single stranded, and it has uracil instead of thymine. Okay. So scientists, you just remember Erwin Chargaff studied amounts of nitrogenous bases in DNA. So he gave the base pair rule. What is the rule that he gave? That A, how much ever is there in double stranded DNA, that much ever T will be there. How much ever G is there in the DNA, that much ever C will be there. That means what? The total number of purines will be equal to total number of pyrimidines. This is applicable in double stranded DNA. Okay. So let's say a DNA molecule, let's say it has total 20% of adenine. Okay. So 20% adenine, we can also expect there will be 20% thymine. Now 20, 20, 40 is done. 60 is left. Out of 60, 30% will be G, and that 30% G, How much is 30% is there? Same amount will be for C also, 30% C. So let's say this entire DNA, it has 1,000 bases, 1,000 base pair you can say, okay, 1,000 base pair. So 1,000 base pair means total 2,000 bases, okay, because each base pair means two bases, 2,000 bases. So how much will be 20% of 2,000 base? So 400 adenine will be there, 400 thymine will be there. Got it? And two hydrogen bonds between them. So total 800 hydrogen bonds between them. Then 30% of 2000 will be 600, 600G and 600C. So between them there will be three hydrogen bonds. Got it? So like that you will have 1800 bonds. So this is how you will have to solve questions regarding this Chargaff's rule. Fine. Then Rosalind Franklin, he gave the X-ray diffraction pattern to see the structure of DNA, and this only cracked the major molecular structure. What Watson and Crick did in 1953, they just published the model. They collected all the information. Actually, they did not do much experiments on their own. It's just that they inferred from the already got results. They just inferred, okay, and they gave the model as a double helix. Last concept about membranes. We'll have to cover this quickly as already our time is over. So I will just go on briefly regarding this. You will be getting notes at the end of this session for the previous biochemistry class also and for this one also. So you can just wait till the end of the class. You will be given a link and you will be getting the notes. So in the notes also I've mentioned all this. You can go through those in detail. Let's quickly cover membranes. Now, membranes are found around each cell, around the cell also, and around the organelles also. Okay, you can see here that these membranes are present all over the cell, and even the organelles, that is mitochondria, vacuole, lysosome, some are even covered by double membrane, like nuclear membrane, mitochondrial membrane, right? So, single membrane can be there, double membrane can be there. Now, what is the design? It is called as fluid mosaic model. Why fluid? Because it is pliable, means it is easily moved. Okay, it is not a rigid structure. It is the molecules can move in that, like how ships will float in ocean. Like that, proteins can also float in the lipid bilayer. Right? Mosaic means what? It's not having any homogeneous structure. It is heterogeneous. It is made up of many different molecules. Lipid will be there, protein will be there, carbohydrate will be there, right? If you notice the carbohydrate components, you can see that carbohydrate components are mostly facing the outside. Why? Because each of them have different function. Carbohydrate on membranes have function mainly for recognition. They will recognize some outer molecules, okay, some plasma membrane of other cell or some matrix components like that. Then phospholipids and proteins make up the most of the membrane. So phospholipids are these ones. You can see structure like this. These are phospholipids. They have a polar head group and a non-polar tail. You understand what is polar, non-polar now. So ones which can interact with water. So that's why they have hydrophilic head. Hydrophilic means they can interact with water. That's why they are facing outside towards the water. But the hydrophobic tails, they are not able to interact with water. They are non-polar. That is why they are sandwiched between the water molecules. Okay, they are hiding from the water molecules on each end. That's why they form a bilayer. Okay, that's why this is a bilayer which is formed. And then in between you have some proteins also. So proteins will be some will be going across the membrane. It will traverse the complete membrane. Those are called as integral protein, or they are also called as transmembrane protein. Trans means they are going through the membrane. Whereas some proteins are there which are present only on the surface. So those are called as peripheral proteins. Okay, they are present at the periphery. Apart from this, you will have some cholesterol molecules also. They are sterol molecules which help with flexibility, and carbohydrate chains which help communicate with other cells. So you must know that membrane is selectively permeable. It means that cell membrane only lets certain substances in and out of the cell. Membranes that allow certain materials to pass through based on certain property. So what property will be there? One is size. Any big molecules cannot pass through the membrane. Hydrophobicity. So our membrane is hydrophobic. So anything hydrophobic can easily pass through it, because non-polar like means uh, the polar likes polar, right? So like charges will attract, isn't it? So this will be hydrophobic, will be interacting with the hydrophobic layer, and they will be able to move through easily, right? So for example, small molecule oxygen can move, benzene it's hydrophobic, it can move, ethanol small molecule it can move, okay? And charge, so charge means if you have any charged molecule, now hydrophobic membrane is hydrophobic, so it will not allow any charged molecule to go through it. Okay, amino acids, which are big molecules, glucose, they are large polar molecules. Amino acid is also a charged molecule. Okay, so it's not that polar molecules can't enter, but if they are small polar molecules, they can enter. But if they are big polar molecules, they can't enter. So this is what is selectively permeable. Okay, semi-permeable, what we can say. So for things which cannot pass, there are some channel proteins, transport proteins. So through that they will pass. That is integral proteins which make up a channel-like structure. Okay. Then you have phospholipid bilayer which forms inside and outside of the layer. They are mostly water. So this is the entire cell. What you have outside is water, inside is water. So lipid bilayers are actually hiding from both the side water molecules. Okay. Bilayer, as I already mentioned, the non-polar tails are facing towards each other. Polar molecules are facing towards the outside. That is water. So in the lipid bilayer, there is nothing non-polar. In fact, even the proteins which pass through the lipid bilayer, you should know that even the proteins which are passing through the lipid bilayer, the portion which is present inside the membrane, that is mostly non-polar. It's made up of non-polar amino acid. The portion which is facing outside, they might contain some polar residues. Okay. So polar heads are hydrophilic, water loving, and fatty acids. Fatty acids means what? It's actually CH bonds, right? There are CC bonds and CH bonds which are there. So they are hydrophobic because CC and CH bonds are non-polar. They cannot interact with water. That's why they face inward. So what are the type of lipids which are found in biological membrane? Many different ones. The lipids which are connected to phosphate group, lipids which are connected to sugar groups, steroids are there, cholesterol, stigmasterol, and glycolipids you have different types, cerebroside, ganglioside, means whether they are connected with two sugars or many sugars, or whether they're connected with some sulfated sugars, right? Sugars can be sulfated also. Phospholipids can be of two types. One in which the backbone of the fatty acid, backbone of the lipid is a glycerol molecule. Those are called as phosphoglycerides, okay, or glycerophospholipids. And the one which will have a sphingosine backbone to them, they are called as sphingospholipids. For example, sphingomyelin. And also we have another lecithin, cephalin, like that. Some things are there. And apart from that, you also have this, as I told you, lecithin, cephalin, phosphatidylcholine, phosphatidylethanolamine. These all will have glycerol as backbone. Okay, phosphatidylserine, threonine, all that. So apart from this, the most important one that will be always present will be phosphatidylcholine. Okay, and phospholipids are the most abundant. The other composition will be less. For example, sphingomyelin you will observe in the myelin sheath of the nerve cells. But apart from that, phospholipids are most abundant. Okay, and they are amphipathic molecules. Remember, so amphipathic molecules means they have both hydrophobic parts and hydrophilic regions. Phospholipids. Let's study the structure of a phospholipid. It's a type of a lipid that has two fatty acid tails. Even amino acids are amphipathic. Okay, so an amino acid, it is called as a Zwitter ion, right? In particular pH, it is called as a Zwitter ion. Why? I just missed out this word. So because it is having, if you have the amino acid in which the amino group is carrying a NH3 plus and it is having COOH, okay. So COH, CO minus, if it is CO minus and NH3 plus, so in that case, yeah, I understand Venkat, but the thing is, I have only 1 hour class and I'm trying to cover as much as possible so that you get, and I have already exceeded 1 hour. That's why I mentioned for membranes I'm going a bit slow. Okay, Venkat, listen to me. Your video will be available in YouTube. Later also you can see this video. Will that be helpful? So once again, later anytime you can pause and you can even control the speed of the video. Okay, you can make it more slower. Playback speed you can make it slower. So even though I'm speaking fast, just to complete this part, you can make my speaking little bit slower. Got it? So later you can again watch this video. Fine, Venkat. I still need to finish some slides and discuss a few questions. So I hope you will understand why I am being a little bit fast. Others, what about others? Are you following? Everyone is following or not? Please, can I have a response? So this is what I mentioned about deuterion, right? So deuterion, carboxyl group, amino group. So both charge will be neutralized. In that case, it will have zero charge. Okay, so if it is zero charge, net charge is zero. Okay, thank you, Indrani, Shubhi, Sharmalina, Jayasavirami, all of you. So net charge if it is zero. Okay, fine. Janvi, Shriram, thank you. Okay, so net charge if it is zero, it will be called as deuterion. Fine. Okay, okay, very good. Shukriya, Vishal, Jay, good. So the phospholipid, actually the structure is like this. You have the glycerol backbone. Okay, so this is the glycerol backbone. Glycerol has three carbon. Over here, three carbon is there, right? So with one carbon, you have a phosphate group, and with other two carbons, you have this fatty acids. This is one fatty acid. This is one fatty acid. So as I said, fatty acids are long CC and CH bonds. They have this non-polar tails. So phosphate head will be considered hydrophilic. In fact, to this phosphate group also some other polar molecules will be attached. Okay, to this also some polar molecules will be attached, like ethanolamine, serine, or something like that. Okay, and the reason why it is hydrophilic is because it is attracted to water. That's why it's considered as polar. Whereas the fatty acid tails, they are hydrophobic because they are repelled by water. So that's why they will be hiding away from the water. This is a phospholipid structure. What you actually draw like this, the structure actually looks like this. The head is the polar group and the tail is the non-polar group. Got it? Then cholesterol, the type of steroid. It's a type of steroid, it's a lipid. It affects the fluidity of the cell membrane. Okay, so fluidity of cell membrane means what? See, in between the lipid molecules, if you have this cholesterol molecules, then what is happening? The interaction between the tails are getting disrupted. Okay, the interaction between the tails are getting disrupted. So in that case, the cholesterol molecules are affecting fluidity. It is increasing the fluidity. Got it? So at higher temperature, what happens to the membrane? Remember, the tails will interact more closer with each other. Okay, they will be more closer with each other. So that is when the membrane will become more solidified. It will stiffen up the membrane. But at lower temperature, because of this presence of cholesterol, it will prevent the membrane from freezing. Okay. Now coming to the proteins. So two type of proteins are there, means function wise. One is recognition, another one is receptor. So recognition proteins, obviously I mentioned that carbohydrates will have recognition function. So proteins that have carbohydrate chains attached with it, they help in cell identification. For example, this kind of acts as a tag, an address tag. Like red blood cells have some specific glycoproteins, some specific sugars. So they can identify viruses as foreign like that. Okay. And receptor proteins are proteins that receive certain chemicals from other cells, as I already mentioned to you before, such as the nervous system cells. Then there are other proteins also, channel proteins. So what will they allow? As I told you, membrane is semi-permeable. So whatever things cannot pass through the membrane directly, they can pass through channels. Proteins that are involved in letting certain substances in and out of cells. One example is aquaporin. Aquaporin is a channel protein which lets water in and out of the cell. Okay, so through that only water molecules can move in bulk. Got it? So membrane basically is a collage of different protein. They are grouped together, embedded in the fluid matrix of the lipid bilayer. So these are, you can see, proteins are folding in the fluid bilayer. And proteins determine most of the membrane specific functions. So you have two type of proteins. One is peripheral protein, another one is integral protein. Peripheral, they are bound to the side surface of the membrane. And integral, they penetrate the hydrophobic core. So they span the entire membrane. Such membranes are also called as transmembrane because they go through the protein, to go through the membrane. An hydrophobic region of an integral protein consists of one or more stretches of nonpolar amino acids. As I said, the one part which is embedded in membrane, that might be consisting of mainly nonpolar amino acids, and they are coiled into alpha helices. Okay. Quickly, what is the function of plasma membrane? Being the cell boundary, it separates cellular materials from external environment. So it doesn't allow everything to come through. It will regulate the materials which can enter and exit the cell. And most importantly, it maintains a homeostasis in the cell. Homeostasis means constant environment. It doesn't let the environment change constantly. Okay. So six major functions of membrane protein will be there. One is transport. So for example, these molecules are coming this side. Okay, sometimes this can be simple diffusion. Through the channel proteins, they will diffuse based on their concentration gradient. Here concentration is more, here concentration is less for the blue molecule. So this is simple diffusion. Okay, but sometimes you might require ATP. Why? Because this is facilitated diffusion, in which you are moving the molecule up the concentration gradient. Here it is less, here it is more. So you need energy to send it uphill. Okay, like that. Some proteins, they will, membrane proteins, they can have some enzymatic property. For example, you have this, I told you last class, you have succinate dehydrogenase. It's actually an enzyme complex which is present in the inner mitochondrial membrane. So what is it doing? It is catalyzing the reaction, that is succinate to fumarate. It is acting as an enzyme. Signaling molecule. So many proteins are receptor molecules. They will have a connection with some proteins in the cytoplasm, and they will have a receiving domain at the extracellular side. So at the extracellular side receiving domain, some signaling molecule will come and bind, because of which the receptor protein undergoes a change in conformation, and that in turn is uh, giving some changes inside the cell. This is what is signal transduction. Cell to cell recognition. You can see two cell membranes are there. So one cell membrane is recognizing the other cell membrane through some sugar parts. Intracellular joining. So two proteins can have direct connection. Okay. So direct connection with which they will join. And last is attachment to cytoskeleton and extracellular matrix. So towards the inside you will see there is attachment with this cytoskeleton actin, and towards the outside you have the attachment with the extracellular matrix components. These are major functions. Okay. So as I already mentioned, hydrophobic polar large and uncharged molecules, they must use a transport protein to enter and exit the cell. The proteins are specific. Glucose can only pass through a glucose transport protein because glucose is a big molecule. It cannot pass through. The rest, smaller ones can, gaseous molecules can. So for special proteins like that, we will require proper protein channels. A special entryway for large polar hydrophilic and charged ions to pass through the cell membrane. So sometimes they will need energy, as I mentioned, facilitated diffusion. Example is sodium potassium pump. For every 2 potassium passed, there will be 3 sodium which is going outside. Okay. So you have different transporter proteins. Protein pumps are there which use energy to increase the concentration on one side of the membrane. We've already talked about this in the last class. So the electron chain complexes, they will transport proteins to the other side of the membrane. So these are called as protein pumps, which will pump against the concentration gradient. Fine. This is acting as a pump. And this is acting as a channel, because we know that if H plus ions go, then ADP and PI is converted into ATP. Okay. So two ways that materials can move in and out of the cell. One is passive transport, active. Passive means what? Without energy requirement. Active means with energy requirement. Passive doesn't require energy means what? It can go downhill from high to low concentration. Active transport means it will go other way, uphill. So it requires energy. Here equilibrium is reached. So normal diffusion happens until and unless everything is equilibrated, this will go around. But concentration become unbalanced because it is going from low to high. So already at one side there is high concentration, but at the same side it will be going. No energy is needed by the cell to move the materials. And here energy is needed. Examples of passive is diffusion, facilitated diffusion and osmosis, whereas active transport works on protein pumps, endocytosis and exocytosis. I'll quickly tell you about endocytosis and exocytosis. Endocytosis means material entered the cell without crossing the membrane. So the membrane just folds like this. Okay, and the particles which are supposed to be taken, they come into a vacuole like structure, vesicle like structure, and they are brought into the cell. This is endo means inside, cyto means cell. Okay, so in endocytosis the cell takes in macromolecules by forming vesicles from the plasma membrane. It's a reversal of exocytosis where molecules will move out. So three types of endocytosis is there. Phagocytosis, phago means some solid eating, cellular eating, so some solid particles it will be taking inside. Pinocytosis is cell drinking, so it will take some smaller particles and same it will be taking inside the cell as vesicles. Receptor mediated means it will have some receptors with which the molecules will bind, then only they can take it inside, otherwise not. Okay. And what is exocytosis? In exocytosis, transport vesicles migrate to the membrane, fuse with it, and release their contents outside. So this is other way around. Some any secretion product, something is produced inside the Golgi complex. So they are sent out in vesicles and they are released outside the plasma membrane. So some secretory vesicles like that. Got it? So this is how the vesicle will fuse with the membrane outside and contents will be released out. So phospholipid, membrane, they will join together. They will integrate into the plasma membrane. Okay. Let's discuss a few questions. Nucleotides have a nitrogenous base attached to the sugar. At which end? So we just discussed, no, this one already. Yeah, very good. Jayasari Ramya, very good. Correct answer. So answer is first carbon, one dashed. Should be right. Very good. It's first carbon. I told you, no, it is connected with one dashed, right? With the nitrogenous base. Vishal, right. Correct answer. Tertiary structure of protein is stabilized various intramolecular intermolecular interaction. Which of the following the weakest interaction involved in stabilization of protein molecules? Out of this, which is the weakest one? Weakest bond. Yeah, Janvi also correct. Very good. Shriram right. Correct answer. No, Indrani, it's not 5 prime. 5 prime carbon, you have the phosphate group attached. Nitrogenous base is connected with the first carbon. Okay. So over here, answer will be Van der Waals interaction, students. Okay, not hydrogen bonds, because compared to Van der Waals interactions, hydrogen bonds are slightly stronger. Covalent bonds are the strongest. Remember that. Okay, and hydrophobic interactions play a major role in stabilizing tertiary structure. So over here, the answer will be Van der Waals. Yes, Jai Sai Ramya and Sriram, very good, correct answer. Van der Waals. Next, molecular weight of arginine is 174 Dalton. What will be the molecular weight of a circular polymer of 38 arginine molecules? So I'll just explain this in case you get confused. So 38 arginine molecules. Now what did I say? It's a circular polymer. Circular means what? There is no free amino or free carboxyl group. That means from each of the arginine you have to remove one water molecule to form the peptide bond, because between two arginine molecule peptide bond will be formed, right? So that's why you have to take 38 into 174. 174 is the molecular weight. Okay, but as we know, when the peptide bond is formed, so this is 6612. As we know, when the peptide bond is formed, water molecule is removed. So again, you have to remove 38 water molecules minus 18 into 38. So that is minus 684. Got it? So 6612 minus 684. 5928 is the correct answer. Clear, everyone? 5928 is the correct answer over here. You have to calculate like this, okay? So circular is given. If it was linear, then you would have said that it is one will be lesser, one less, okay? If it was linear, fine. The next one, in the plasma membrane, the glycolipids are located at. Okay, so Priya, I think you got it. It is Van der Waals force, okay? Yeah, very good, Shubhi, correct answer. Jai Sai Ramya, correct answer. Very good. So answer is outer layer, okay? Because they have to interact with the plasma membrane of other cells. They have to interact with the matrix of other cells. Means matrix component. Yes, Janvi also right. Very good. So they have to be oriented towards outside because the main function of sugar, we are talking about glyco. Glyco means sugar. So sugar's main function is for recognition, and recognition will happen only on the outside. That's why one is the right answer. Last question. The nature of the peptide bond is that of. So I already mentioned peptide bond formed between two amino acids. Yes, very good. Jai Sai Ramya, very good. The answer is amide linkage. Yes, Vishal also right. Yeah, Sharmanina right for the previous question. Okay, yes, Janvi right. Answer is amide linkage. Very good, students. Thank you for paying attention. That's all for this class. You can get the link from the chat box. You will be getting the link of the notes. So you can download some notes of this, whatever I taught you. All the best to you for your exams. Do well, study well. Thank you. Good night.